Hello, can you all hear me? Perfect. That's a fantastic way to introduce this topic as a topical one and borderline controversial one. Myrtle rust. Myrtle rust is something that's come into the country in May 2017. It's now sort of spread throughout the North Island and the top of the South Island. But what actually is myrtle rust? It's a fungal pathogen that targets plants in the Myrtaceae family. So the plants that are going to be of interest to some of the audience here are your manuka, your kanuka, rata, and my favourite tasting honey, which is the pōhutukawa. But it's not just these four plants that are going to be targeted, it's potentially the whole Myrtaceae family as well. Myrtle rust works by affecting the new vegetation and the new growth on plants. It targets the new growth and forms these rusty pustules, and eventually kills them off. And so you don't necessarily see massive damage and massive plant loss initially. It can take quite a few years to see dramatic losses in plants and vegetation cover. And so you can see here, the rust can get pretty intense. And these photos were taken in Brisbane, where we did our field work. As you can see, it can cause quite extensive damage and end up killing the leaves entirely. And so this project all took place in Brisbane. You can see here there's native bees over in Brisbane actively collecting the rust spores from plants. And the rust not only occurs on the leaves of the vegetation, but it can affect the flowers as well. So you've got the rust growing on the buds there, and it can deform the flowers. So it's on the leaves, on the flowers, and it's all in close proximity. So it's not unreasonable to expect bees to not only actively collect it, but to passively pick it up while they're out foraging. And there's another photo there to demonstrate that. This is an Australian species of Leptospermum covered in the rust spores, and so we expect that this might be taking place here as well eventually. And this photo here was taken in Moggle, which is about 3 hours out of Brisbane. And that tree there you can see has massive losses in vegetation cover, and it's almost completely died from it. So our project, in short, we placed a bunch of beehives out in areas that we knew there were lots of myrtle rust spores, and we left them there for 2 weeks for bees to go out and forage and collect as much in as they possibly can. We blocked off the hives and collected the forages as they were coming back in, and we collected bees from inside the hives, and we scooped out pollen cells as well, and we collected all that up. We then moved these hives into containment sheds, which we've got an example there, and we kept those bees alive in there for up to 2 weeks. And on days 0, which is the day we put them in, the first day, day 4, 7, and 9, we went through and we collected bees. We put bees in cages with myrtle rust on them, and we put rust spores in these little tubes, and we put those in cages as well, and sampled those on those days before to get a measure of spore viability, how well the spores can remain active and viable inside those hives. And this here is a first graph to show you. This is showing you on the left-hand side the returning forages, so the bees that we were collecting as they returned to the hives out in the field. And you can see that those ones, the bees coming back, they were carrying myrtle rust spores on them. Whether they had been actively collecting them or not, or picking them up, we don't really know that one yet. And the bees inside the hives, which is that middle figure, you can see that bees inside the hives were also carrying rust spores. And the same with the pollen cells. So we scooped those out, did some genetic tests to look at the presence of myrtle rust spores, and at almost every single site we had hives at, rust spores were being packed into those pollen cells. And so that leads us to the question about spore viability. So once they're back in the hive, how long do the spores remain viable for? You can see over that 9-day period, there is a gradual decline. The evidence for that is rather limited, so we need to do a lot more work into it. But at the end of 9 days, you can see viability remains about 15%. So about 15% of the spores can remain viable and will germinate. That's the sort of standard level that people have been getting when they look at myrtle rust. So seeing those high numbers in those first few days is quite alarming and quite outstanding as well. That leads us on to quite a few more important questions as well. We don't actually know how much of a problem this is in New Zealand. We don't fully understand how often bees are bringing rust back into the hive, so we still need to find out if they're actively collecting the spores or if they actually are just passively picking them up. But we also want to know what drives bees into collecting these spores when they do. Is it down to poor resource availability, or is it just it's there, they do it because they get confused, or we don't really know. But the other important thing that we need to find out is, are spores leaving the hives? So we know they're coming in and they remain viable, but are spores then leaving the hives and able to reinfect plants? That's the next sort of question, sort of next areas of research we're hoping to look in doing. And you can see there in that last photo, there's a nice wee Australian bee posing quite nicely on a bit of myrtle rust there. But that brings me to the end of my talk, and I've got some time for questions.